so welcome back everyone. We're going to continue our discussion now of genetic variations with specific focus on one of the main reasons people look at genetic variation data, and that's to find correlations between genotype and phenotype. And phenotype very often will refer to disease phenotype, so trying to find genetic variations that are associated with risk of disease. And what we're going to talk about today is primarily some of the statistical and informatics methods that go into this, with a focus on some of the more modern methods for working with relatively large data sets. So just to review from last time, we had looked at some of the basic issues in genetic polymorphisms and phenotypes. So we mentioned that we were often interested in discovering variants because they're associated with a phenotype. And in some cases, that's going to be because a variant directly causes a phenotype, such as a single base change causing sickle cell anemia. But very often, what we're really going to be looking for is something that is a little easier to find, and that is variants statistically associated with a phenotype. So variants that occur more often in people who have a particular phenotype than people who don't, maybe not because they're directly causing the phenotype, but because they're correlated with a causal variant or variants, often because of physical proximity on the genome. And we're going to look at some of the basic methods for doing this. So one of the big issues you need to consider when talking about this question is the issue of study design. So study design refers to how you plan gathering your data to give you a reasonable chance of getting these kinds of correlations and discovering them from statistical analysis of your data. Classically, the way this was done was usually with linkage studies until relatively recently. Linkage studies are also known as pedigree studies, and they follow a relatively simple general idea. The details can get quite complicated, and I'm not going to go into all of them, but I think it's good to be aware of what a linkage or pedigree study is, and the general ideas it uses, and the situations in which it's appropriate. So a pedigree study follows from a pedigree, that is a family history, in which we have at least a reasonable fraction of the people annotated based on whether or not they have the phenotype or the disease of interest to us. So maybe we've got a family where a large fraction of the people come down with colon cancer, and we might be interested in trying to find genetic variants that predict your risk of colon cancer. So we would have a family tree in which we have different individuals. So this would represent a father and a mother. These would be their children. And we would have here a code that tells us that some of the individuals are affected. So affected would mean they have the phenotype, say colon cancer, and some of them are unaffected, meaning they do not have the phenotype. And we would hope that we would get a few generations of this, at least a few generations of people in which we mostly have this kind of information. In a linkage study, we would then type a set of markers in these people. So we would try to figure out genetic variations at a small set of sites in the genome or a region of genome of interest to us. And these might be sites where we have two possible alleles. They could be multiple alleles, but generally a small number of possible alleles at each variant site. So we might have a marker site A where we have alleles big A and little a, marker site B where we have alleles big B and little b, and marker site C where we have alleles big C and little c. And these will typically be spaced on the order of megabases apart on the genome, so millions of bases apart from one another. And as we'll see shortly, that's an important feature and an important limitation of these kinds of studies, that these are typically millions of bases apart from one another. But we will type these variants, and we will try to figure out which combination of alleles each of the people in our study has by a genetic assessment of the genotypes of these people. There is a complication here I'm kind of glossing over, which is that usually you can't easily figure out which of the two alleles is on which chromosome, but let's just pretend for the moment we have a way of figuring out that this individual here had one chromosome with alleles big A, little b, big C, and one chromosome with alleles little a, big B, little c, and this other individual here, who was a mate of this person, so they had children together, had one chromosome with alleles big A, big B, big C, and one chromosome with alleles little a, little b, little c. So we would try to figure this out for all of our individuals. It's very likely we won't be able to find it for all of them because there's a pretty good chance some of these people are not alive anymore, or not available, or not willing to participate in the study, but there are ways of inferring what these values are if you don't have them for everyone. everyone. So more or less you can come up with this model of which pieces of chromosome or which combinations of alleles are found in each individual. And the basic idea behind what we're trying to do is to identify pieces of chromosome that generally pass together with the phenotype. So, for example, we might observe that this combination of alleles, big A, little b, seems to, in this pedigree, consistently pass along with the phenotype. Affected individuals seem to consistently inherit that little piece of chromosome starting from this founder individual in the first generation of our pedigree, and then that piece of chromosome passes down to different individuals. Often in practice, it's not going to be quite that clean. You won't necessarily have a single unique piece of DNA that's found in everyone who's affected, not found in everyone who isn't affected. It may be, if we're talking about a complex disease like colorectal cancer that has a lot of genetic and environmental influences, that some of the people who don't get that piece actually have the disease, some people who do get it don't have the disease. But at a high level, let's say we can detect this strong correlation between that piece of chromosome and the disease phenotype. That would then suggest that the actual disease-causing variant is probably more or less in that region of genome, or at least close to it. We wouldn't see that kind of correlation, most likely, if the disease-causing variant that was raising risk in this family wasn't found on that piece of chromosome. And figuring that out is the general idea behind a linkage study. Linkage studies depend on a very basic property of eukaryotic reproduction, and that is the process of meiosis, and in particular, the process of recombination that occurs during meiosis. So when two individuals from a eukaryotic species like us reproduce, for the most part, our DNA is contained in autosomal chromosomes. We have pairs of chromosomes, one inherited from the father, that's the maternal, a maternal inherited from the mother, and then the child of this person and this person would inherit one chromosome from each parent. So that person in the child generation would have a paternal and a maternal chromosome. But the really important point for a linkage study is that the paternal chromosome this individual inherits from the father is not exactly either of the chromosomes the father had. It's going to be a hybrid of these because of the process of recombination. So what recombination does is create a new gamete, so a new version of this chromosome, that's based on stitching together pieces of the father's paternal and maternal chromosomes. So what the child inherits as a paternal chromosome will be a kind of patchwork of pieces of the father's maternal and paternal chromosomes, and likewise for the mother. So the child will inherit a maternal chromosome that is pieces of the mother's paternal and maternal chromosomes. And this patchwork will generally have a resolution on the order of tens of millions of bases between switches of the chromosomes. So you might get a few tens of millions of bases of the father's paternal chromosome, then switch to material from the father's maternal, maybe switch back to material from the father's paternal again, maybe do this a few times. But basically you're going to get that kind of breaking up of the chromosome on a resolution of tens of megabases typically. And that's really important because if you didn't have this recombination process, linkage studies wouldn't work. We would never be able to figure out where within the genome the disease-causing variant was if we didn't have this kind of breakdown into these smaller pieces, because if the disease-causing variant happened to be on, say, the father's paternal chromosome, then that would be handed down intact to everyone who has that variant, and there would be no way to tell whether the variant was on one part of the chromosome or another. The entire chromosome would keep getting inherited intact with that variant. So we need this process, and it's important that this process is a relatively coarse-grained thing in terms of genomic scale. So it's important that this happens on this megabase scale because it means that in a relatively large pedigree, so maybe you can get to 3, 4 generations, you're not going to have a resolution of these segments of more than a few megabases. And so there's no hope of tracking down where the variant is to a finer resolution of more than a few megabases. So that is a key limitation of these studies, and it's an important thing to be aware of. So linkage studies have been used very effectively. They've been the basis of discovering quite a lot of genotype-phenotype correlations, but they do have these limitations. One of the obvious ones is that you need a pedigree, and that means that you need an affected family. So if you're trying to study the genetic basis of heart disease by a linkage study, you have to find a family that has a high incidence of heart disease who has a chance of finding this variant. That family needs to have more or less the same genetic basis for their disease as the general patient population, maybe not exactly the same alleles, but at least more or less the same genes implicated, or else you're not going to learn anything that is relevant when you are outside this family. Now, maybe it's worth doing to help one family, but usually we're trying to do these kinds of studies to find the genes that are implicated in a particular disease more broadly, so that we can then do further study, find out exactly what causes this, and maybe develop therapeutics, diagnostics, treatments of various kinds. So this is often going to be a limitation. Some diseases you'll be able to find a family that has a trait like this, some diseases you won't. It's also a problem that when you're dealing with families like this, with rare exceptions, you're not going to get that many people or that many generations. And because of this, you need a disease that has what's called a very high penetrance. So penetrance of the phenotype, we would say, or the genotype, refers to the probability that the genotype is in concordance with the phenotype. So a genetic variant with high penetrance would mean that individuals who have the variant get the disease, individuals who don't have the variant don't get the disease most of the time. A lot of what we're interested in studies like this today is finding low penetrance variants, the ones that maybe individually change your risk just a little bit, but accumulated over many of these variants might be important predictors of whether you're at particular risk for a disease. And again, with rare exceptions, you're not going to be able to find those with these linkage studies. Finally, because you need recombination between the markers, there's no value in having a marker spacing of finer than a few megabases. So in other words, you want your markers to be spaced megabases apart, and if they're closer than that, you're not going to be able to resolve which of the markers is associated with the disease. That means you're not going to be able to pin down very exactly where these traits are, where the variants that are associated with the phenotype are. You might be able to get within more or less a region of a chromosome and then bring in additional biological knowledge and further studies to refine it more finely and maybe pin it down to a particular gene. But that's going to be a big undertaking, and it is another important limitation of these linkage studies. So because of these limitations, a lot of the field moved to a pretty large degree to a different kind of study design called an association study or case control association study that tends to be more suited to the modern kinds of big data sets we heard about last time. So the detailed, finely mapped SNP or other localized variation data sets where we might be able to look at millions of genetic variations across an individual's genome. In a case control association study, what we would look for typically are a very large number of markers, and in principle these can be very densely spaced, even kilobases, so thousands of bases apart, so much more finely spaced than would be possible with a linkage study. And what we try to do is put together two populations of individuals. Instead of looking for related people, we would typically look for specifically unrelated people. And there are some exceptions where you look for particular kinds of relationships, but in the general case, we are looking for unrelated individuals. And we have one population known as the cases. So these are individuals who have the phenotype of interest to us, let's say colorectal cancer. And then we put together a control population, so individuals who don't have the phenotype of interest. You need to be quite careful in assembling these populations to do what's called demographic matching, basically make sure that in every way other than the phenotype, these populations are as similar to one another as possible. Otherwise, you can get distracted by things that incidentally correlate with the phenotype. But basically, if you do a good job of this, you can assume that you've got a large set of cases and a large set of controls who are more or less random individuals from whatever population you've sampled, and who are distinguished in principle only by the fact that these have the phenotype of interest and these don't. And what we then try to do is identify specific genetic variants that are associated with the phenotype. So in the simplest version, we would run through individual genetic variation sites until we find a site like maybe this fourth SNP here, where we can see that there's a significant imbalance in allele frequencies between cases and controls. So the cases here we can see usually have an A allele at this site, the controls usually have a T allele, and if we can find a strong enough bias like this that we're convinced it didn't occur by random chance, then we would say that we seem to have pinned down on a variant that is statistically associated with the phenotype. Because of the way this design works, we are likely, or at least have a chance, of finding a variant that is much closer to the actual disease-causing variant, maybe actually is the disease-causing variant, than you're likely to get with a linkage study design. But we would generally assume that this is statistically associated, may or may not be the actual disease-causing variant. Now that's the basic intuition, but if we want to use this, we need to go a bit beyond the intuition and know something about the techniques behind this. And this primarily comes down, at least in the way this is usually done, to a statistical association problem. So statistical testing to figure out whether we're seeing an allele imbalance at a particular genetic site in cases versus controls. That's commonly done by first putting together what's called a contingency table. So a contingency table is simply a way of organizing the information about cases and controls and allele values at a particular site that will then feed into the statistical test we want to do at that site. So in a standard 2x2 contingency table, what we will do is take two different variables of interest to us. And the variables of interest to us are the case control status and the allele value at a particular site at which we're looking at that moment. So maybe we're looking at a site that has two allele values, a C or a G, and we would look at how many individuals in the case population have a C at that site, and we would put that in one element of the contingency table. So let's say 150 of the cases have a C, and we would look at how many of our cases have a G, and maybe we observe 650 cases have a G, put that in that site. Likewise, we would look at the controls. We might observe that 200 of our controls have a C at that particular site, and 600 of our controls have a G at that site. So those four numbers would then become the elements of our contingency table. And what we're trying to ask in a more formal sense is, can we reject the hypothesis that the variable of case control status is independent from the variable of allele status? In a sense, what we're trying to ask is, if you fix one of these variables, is the other distributed independently of the variable you fixed? So in other words, if we look at the cases versus the controls, do we see the same frequency of C's versus G's in the cases as we do in the controls? Now, when you're working with finite amounts of data, so finite numbers of individuals, just by random chance, there's going to be some skew in those numbers. So we really need to see large enough differences that we have very convincing evidence that these are not the same distributions. They're not drawn from the same distributions. So here we might say that the allele frequency of C versus G in the cases is 150 over 150 plus 650, so 150 over 800. The allele frequency in the controls is 200 over 200 plus 600, so 200 over 800. 150 over 800 is not that different from 200 over 800, so we might say that this doesn't look like evidence of association, at least not convincing evidence. In this case, we might see much stronger evidence of a skew, so we might have another site where the alleles are A versus T, and we might see that the cases have a frequency of 800 out of 900 A's, whereas the controls have 200 out of 900, and we might say intuitively that that looks like a pretty strong skew. That looks like strong evidence that these two variables are not independent, and therefore that this allele value is in some sense predictive of or associated with case control status, and maybe a hint that that SNP is one that might be relevant to the disease, or at least close to a site in a particular gene that is relevant to the disease. So that's more or less the intuition, but we need to go beyond intuition. In this context, they're worth knowing about because they come up again and again in various other contexts we're likely to encounter. And for contingency tables, Generally, the most common thing we'll use is what's called a Chi-Squared Statistic, which is simply a standard way of statistically testing whether the two categories in a contingency table are independent of one another. So there are other tests you'd use in specialized cases, such as if the numbers were very small here, but the Chi-Squared is more or less the standard thing we would use. So I want to go through a bit how you would do a Chi-Squared test. And it's not really that complicated, but the formula can be a little confusing if you haven't seen it before. The first thing we would do is we would fill in our contingency table, and we would then add up the rows and columns and add up the total numbers in the table. So in other words, we see that we have in the A allele 800 cases, 200 controls, so we would say we have 1,000 individuals with the A allele, adding up those two values to get the 1,000 on the right here. For T, we have 100 cases, 700 controls, we have 800 individuals, and we accumulate the cases. We see we have 800 plus 100, that's 900, and controls 200 plus 700 is 900. And then if we add either of these totals, 900 plus 900 or 1,000 plus 800, we can see that we have a total of 1,800 individuals in this contingency table. And those are the numbers we're going to need to figure out whether this is a significant value. So to compute the Chi-Squared statistic that we're going to use to test for significance of this contingency table. The null hypothesis we're going to use is that these are independent random variables, and the statistic then, the value we're going to compute, whose p-value is going to tell us whether we can reject the null hypothesis, is given by this formula here. So we add over cells of the table the quantity expected minus observed squared over expected. So expected refers to the number a particular cell in the contingency table would have if the null hypothesis were true. So the way we would figure that out is we would look at the two variables independently and figure out what fraction of individuals should have allele A, independent of case control status, what fraction of individuals should be cases, independent of allele status, and then the total number of expected individuals in a cell would be the fraction of individuals who should have the corresponding allele value times the fraction who should have the corresponding case control value times the total number of people in the table. So in other words, if we take this first cell here, we would say that we first want to look at the fraction of individuals who are cases. We have 900 cases out of 1,800 people, so 900 over 1,800, or 1/2 of the people should be cases. We multiply that by the fraction of people who should have an A allele. We can see that we have 1,000 individuals with A alleles, 800 individuals with T alleles, so we get 1,000 over 1,800, so 1,000 over the total number of people. Fraction of the people should have an A allele, independent of case control status. And then we multiply those two fractions by 1,800, and that is the expected number of individuals who would have both the A allele and the case status, and so it would be in this upper left cell of the table if the null hypothesis were true. So 900 over 1,800, that's 1/2. 1,000 over 1,800, that would be 5/9. And then we multiply that by 1,800, and we are going to get our expected value. We then subtract from that the observed value, which is simply the number actually in that cell of the table, square that, divide it by the expected value, and that gives us the contribution to the Chi-Squared score for that element of the table, that cell. We would repeat that for each of the cells. So we could say with the upper right cell, we would ask what fraction of individuals are controls. That would be 900 over 1,800. What fraction of individuals have the A allele? That would be 1,000 over 1,800. Multiply that by 1,800. That is the expected number in this cell. We subtract 200, square that, divide it by 200. Then that gives us the next contribution, and likewise for the two cells corresponding to the T allele. Add those up, and that is our Chi-Squared score. So that's how you compute the Chi-Squared statistic. So under the null hypothesis, what we would expect from that formula is that you are going to get values close to 0 if the null hypothesis is true. So in other words, if the null hypothesis is true, What that is saying is that the observed values in the table should be close to the expected values. That's what the null hypothesis asserts. So expected minus observed should be around 0. Expected minus observed squared over expected should be around 0. You add that up over the cells, you should get a small number. Not exactly 0, but close to 0. And if the values are correlated, however, you're going to see that skew in the table. You're going to see differences between expected and observed values. Some will be observed in the direction of being higher than expected, some lower than expected, but it's likely that you're going to see values skewed away from 0. So when you take observed or expected minus observed, square that, you're going to get a positive number, and generally a relatively large one if there's a large skew. So what you're looking for then is that skew, relatively large chi-squared values, to assign a p-value to your chi-squared statistic. Now this statistic is a little more complicated than, let's say, the binomial random variable that we've seen before. So there isn't a simple formula we can show to compute the p-value, but the math can be worked out. You can write a computer program. It's a little more advanced than we cover in this class, but you should be aware that this is doable. You can look up tables of these, and now it's very easy to get chi-squared calculators where you can plug in your chi-squared score or just plug in your contingency table, and it will do all the computations and tell you the p-value for your chi-squared score. So basically that gives us a way of getting p-values and figuring out whether we can definitively reject the null hypothesis. So that brings us back then to the case control study. Basically what we are doing is simply running through our allele values, running through our SNP sites, and performing these chi-squared tests one site at a time. So we could look at this first site here. We could see there are A and G alleles. We can look at our cases and controls, and we could then go through and do this exercise. Usually we wouldn't do this manually. We'd write a computer program, but more or less what we could do is take those values. We can say that there is some set of cases, some set of controls. If we're looking at this particular site, we can say there's some of the A allele, some of the G allele, and we could count. If this were our entire table, what we would find is that we have 3 cases with the A allele, 2 cases with the G allele, 3 controls with the A allele, 2 controls with the G allele, and we would then compute our chi-square scores for this table. So if we wanted to do that, we would sum up our values here. We've got 6 instances of the A allele, 4 of the G allele, 5 cases, 5 controls, so a total of 10 individuals in our table. And we could then start computing those chi-squared scores, the contributions for each of the cells. We could look at our first cell here. We could see that the observed value is 3. We could try to compute an expected value. So we would take the fraction of cases, let's say. We have 5 out of 10 individuals are cases, so 1/2. We could take the fraction of A alleles. We can see that 6 out of 10 individuals have an A allele. And we would multiply that by the total number of individuals. That's 10. And we could then observe that the expected value in that particular cell of the table is 5/10 times 6/10 times 10. It's 30/10 or 3. And we then subtract from that the observed value, that's 3. So we're getting 3 minus 3 squared, and divide that by 3, which is simply this thing. And what we're going to end up getting out of that is actually 0. The expected value is 3, the observed value is 3. 3 minus 3 is 0, square of 0 is 0, 0 divided by 3 is 0, so we get a 0 out of that. If we repeat that for the other cells, we would get same thing for control and A. 5/10 of the individuals are controls, 6/10 have A, multiplied by 10. We get an expected number, 3 of controls with the A allele. 3 minus 3 is 0, 0 squared is 0, 0 divided by 3 is 0. And a similar thing would occur for the other cells in the table. So if we take the expected number of individuals in this cell of the table, we would take the fraction of cases, that's 5/10, times the fraction of G's, that is 4/10, times the total number of individuals, that is 10. We would we subtract the observed value, which is 2, square that, divided by the expected value, which we just figured out is 2, we get 0, and we would end up with the same numbers from the last cell. So 5/10 is the fraction of controls, times 4/10 is the fraction of g's, times 10 gives us 2, minus 2 squared over 2 gives us 0. So if we go through the whole exercise here, we would actually conclude that the chi-squared score for this first SNP is 0. So that we would conclude is going to be not a significant value. We are looking at one-sided p-values here. We're looking specifically for skew in the positive direction. So seeing a 0 here is going to tell us that we're not looking at a significant association. We could keep going through our data set. We could try another one. I'm not going to go through the exercise of doing all of these, but we could compute another value here, or maybe this one's a little more skewed. So 3 C's in controls versus 2 C's, or 3 C's in cases versus 2 C's in controls, and so forth. But eventually we would hope that in running through this, we would get to something where we would see a strong skew, we would get a large chi-squared score, and we could compute a p-value that is very low and allows us to say that we are looking at a statistically significant variation. Right, so there are some appreciable problems with this. And probably the biggest one to deal with in practice is the issue of multiple hypothesis correction. So generally when we're doing this, we're not looking at just one SNP. We're generally looking at many SNPs. In a large study, so a genome-wide study, we might be looking potentially at hundreds of thousands or millions of SNPs. And because of this, we have to worry about the problem of multiple hypothesis correction. If we're looking at 1 million SNPs, even a p-value of 1 in 1 million is not going to be significant because you would expect something on that order by random chance, even if the null hypothesis were true if you're doing this trial 1 million times independently. So we have to do a correction for p-values. This particular field does something similar to the e-value correction we saw when looking at BLAST and sequence alignment, but it's a slightly different strategy called Bonferroni correction, where basically what we try to do is get a corrected p-value that adjusts for the fact that we're doing this multiple hypothesis correction. As long as we're looking at relatively small p-values, you can approximately get the Bonferroni corrected value by multiplying the number of tests you're doing by the p-value, more or less the same way we would get an expectation or an e-score, or other tests. This doesn't work if you are looking at relatively large p-values relative to the number of SNPs. More or less, this formula is going to be accurate as long as the product is significantly smaller than 1. But we're only interested in SNPs where the corrected p-value is significantly smaller than 1, so that's not really a big limitation. In practice, what this means is that we more or less take the p-value we're trying to get. So let's say 10 to the minus third we might decide is a significant p-value. That would be a typical cutoff for this field. And then you divide that by the number of variants you're looking at to get the corrected p-value you need on each individual variant. So if you want an overall p-value of 0.001 for your study, 10 to the minus third, and you're looking at 1 million SNPs, the way you would compute that p-value is to divide the p-value you want by the number of SNPs, so 10 to the minus third divided by 10 to the sixth, and say that you need a p-value of 10 to the minus ninth on an individual SNP before you'll believe that SNP after multiple hypothesis correction. So that basically is how we would generally do these kinds of studies. There are some other techniques you'll see. So there's a newer one that's often used now called false discovery rate, or FDR. It's a slightly different way of thinking about multiple hypothesis correction that has some advantages in pulling out the kind of weak effects in some kinds of study designs. But for the purposes of this class, just be aware that multiple hypothesis correction is an important issue, and Bonferroni correction is a common way that's handled. There are a lot of variations on this that are often trying to deal with this multiple hypothesis correction problem in various ways. So one way of dealing with this is to try to cut down on the number of variants you're looking at by using combinations of variants called haplotypes instead of individual variants to do these tests. So very often, because of the process of recombination, over relatively small numbers of generations, and human beings haven't been around for that many generations, you would find pretty strong correlations between nearby SNPs on the genome. This is sometimes referred to as linkage disequilibrium, but basically within small regions of the genome, even though you might have many different genetic sites, many different variant sites with their own allele values, you will often find that the sites are highly correlated, and you only have a few global patterns. So maybe you have 3 sites, each of which has 2 values in this region. In principle, that would give you 8 possible combinations of allele values across the 3 sites, but it might be that you only observe 2 or 3 of these because they're strongly correlated. So those combinations are known as haplotypes, and you can generalize the kind of testing we were doing to do the statistical test where your alleles are the haplotypes instead of individual SNPs. If these are strongly correlated, so you don't have that many of these, you can get effectively a stronger test because you are not correcting as much for multiple hypotheses. You lose a little statistical power by having more categories in your alleles here, but you more than gain it back by having many fewer tests you need to do if you have reasonably large haplotypes and not too much diversity within the region of these haplotypes. So that is one way this is commonly improved upon. You can also play around with ideas of trying to limit the number of variants you're doing. Very often what people are trying to do in this kind of study today is what's called a genome-wide association study, often abbreviated as a GWAS study. A lot of people are interested in GWAS because for many of the diseases that we're trying to study genetically, we really don't know what's important genetically to contributing to those diseases. Things like cancer risk or heart disease or diabetes, the so-called complex diseases, are situations where we know there is a strong genetic component in who gets the disease, but we can't find too many very high-risk individual alleles. So these are called the complex diseases, and what's believed is that large numbers of alleles each contribute a small amount to your risk of getting those diseases, and in situations like that, we want to cast a very wide net. We want to look across the entire genome because we don't know what's important. But that creates a very strong multiple hypothesis correction problem. You need very strong p-values, very small p-values for each individual site to get significance. So you would often balance that with what's called a candidate gene or other kinds of pruning strategies, where basically you try to focus your search on things you think are more likely to be important. It may mean that you are looking at a small number of genes or genetic regions where you have prior reason to believe there's something important going on. Very often this is done at the level of variance, so trying to prioritize to a small number of SNPs that are in regions of the genome you think are more likely to create an effect. So maybe you think that there's a high probability that variants that sit in important regulatory regions of genes are ones that cause diseases, then you might only focus on those SNPs as another way of cutting down on the number of tests you do and therefore having a less aggressive multiple hypothesis correction. The current state of the art is advancing quite rapidly in this area, though. There's a lot of interest in doing these genome-wide studies. Many have been conducted, and a lot more effort is going on continuously to gather these data sets. And as it gets easier to get more and more data, to gather larger and larger data sets on more and more diverse populations. A lot of people think that this is the best option available for getting at the genetic basis of complex diseases, and there have been a number of important successes, but it is worth knowing that this is a somewhat controversial hypothesis. There's a lot of dissent in the field about how well this is going to work. The basic problem behind this, or the theory behind why there's a problem, is that this is going to work well if you have not too many variants and each variant has a relatively large contribution to the risk of the disease. The concern there is that there is no really solid evidence that that's the case, and this will not work if we have a huge number of variants, each of which contributes a very tiny effect on the risk of the disease. So if there are almost all of them contribute a very tiny fraction to your risk, then this kind of study design is not going to work. But basically, People are gathering the data on larger and larger scales, pushing ahead, even though this is maybe an unresolved question. There's a lot of effort on both the statistical side and the data gathering side to at least do the best we can with this and try to resolve this controversy. A lot of the work that's going on in the field has to do with controlling for exactly this problem of multiple hypothesis correction. So trying to come up with better ways of controlling for multiple hypotheses and using some more advanced techniques that I've discussed here to try to group things more carefully or better weight certain SNPs versus others to try to do this kind of control. There are also other issues I haven't really gone into here that are getting a lot of attention in the literature, such as control of demography. This is a very hard problem to handle what are effectively unintended effects, unintended correlations that arise because of common ancestry. So which people are more closely related than other people. This is a difficult problem to solve, and many people in the field are working on this. But that is more or less where this particular field stands today. It's a very active research area from both the data gathering and the informatics perspectives. A lot of people have hope that we're going to be seeing much more interesting things coming out of this, continual flood of new discoveries. That is slowly coming out, but still controversial how well this will work in the end. I want to talk before we break about one more kind of genetic variation study that people often do that's closely related to this, but uses somewhat different statistical methods to answer a somewhat different problem. And that is called quantitative trait locus, or quantitative trait loci studies, often abbreviated QTLs. So a QTL is another kind of genetic variation that correlates with a phenotype. And the distinction between a QTL and the kinds of variants I was talking about previously is that a QTL has a real valued phenotype. So that is a number that varies over some continuous range. So we were previously seeing what are sometimes called categorical phenotypes. So either you have a disease or you don't have the disease. In a QTL, we're interested in a trait that would have a value. So it might be that weight is a trait that we're interested in. So it may be that we're looking for variants that associate with weight. A weight would be a continuous number then. And very often this is something that's used more in agricultural contexts, at least traditionally, than in human medicine context or human research context, although that's changed in the recent years. It has some important uses in human medicine. But let's just say that we're looking at the canonical case of an agricultural application. So maybe we are farmers, we are trying to grow corn, and we're interested in the weight of yield of corn from plants based on genetic variations. We'd like to figure out which plants have genetic variations that bias them to producing a larger weight of corn so that we can selectively breed those plants and come up with better, higher producing strains. So we can do something very similar to our association studies for categorical traits, genotyping at a large number of SNP sites within our corn plants, say, and looking at a set of these corn plants and seeing what weight of corn we get off of these plants. So we get our trait value, our quantitative trait, and what we're trying to do then is something qualitatively similar to what we want to do in the categorical tests. We're trying to find genetic variants that associate with the phenotype of interest. But in this case, we're trying to find variants that associate with the quantitative value here. So, for example, to identify that this site, which sometimes has C alleles and sometimes has G alleles, is associated on average with a higher weight of yield of corn plants. So that is the task in quantitative trait locus analysis. Now you could turn this into something you could study with the same kinds of methods we saw for categorical traits by turning this into a categorical trait. So we could put a cutoff here and say that we will say that plants with a yield of, or weight above 6 have high weight, and plants with a yield below 6 have low weight, and then treat this as if it were a categorical or binary phenotype. C's here would be associated with high weight, G's with low weight. But that's throwing out a lot of useful information we get from variation within the values, within those categories, and so we often want to do something a bit more intelligent there. So generally what we would want to use is a different kind of statistic that is designed specifically to work with this kind of quantitative data. And the standard way we would do this is by trying to pose the problem of whether the values we see associated with one allele seem to be drawn from a different probability distribution than the values we see associated with the other allele. We could pose that as a statistical null hypothesis by saying that the null hypothesis is that the values associated with C allele, let's say 1087, are drawn from the same probability distribution as the values associated with the G allele, let's say 5, 3, and 1. The most common way to do this is to assert that we believe these values are drawn from a normal random variable or a Gaussian. It's a kind of a very standard random variable that comes up in a lot of kinds of analyses. And if you make that assumption, then you can end up with a statistic called a student's t-test, which gives it an effective way of figuring out if you can definitively reject that null hypothesis. The t-test comes in a lot of different variants. This is in some sense the most generic of these, but gives a good model we can use to understand, at least intuitively, what a t-test is doing and when we would want to use it. So the basic idea behind a t-test is that we are going to look at the ratio of two different quantities. One of these quantities is simply the difference between the mean values in the groups. So if we look at the average value in the group of plants that are associated with the C allele, and compare that to the average value in plants associated with the G allele, then we would expect that those averages should be very different if there is an association between this allele and the phenotype. So if the C allele, let's say, produces higher yielding plants, you would expect the mean of group 1, that is plants with the C allele, to be noticeably larger than the mean of group 2, plants with the G allele. So that is the numerator of the statistic we compute in the t-test. You have to control for the fact, though, that how important this is, or how big that skew is going to be relative to how spread out the values are. And that's captured in the denominator of the t-test, which, again, this differs between different versions of the t-test, but roughly speaking, what this is computing is approximately the standard deviation, which is a measure of spread of the values within each of the categories. In this version of this, basically what we're doing is averaging the variances, which is the square root of standard deviation of the two categories, taking the square root of that, and getting something, as I say, more or less a measure of how spread out the values are within the individual categories. So if the means of the categories are far apart relative to the average spread within the categories, then you would say it looks like these are drawn from different probability distributions. If the means are close together relative to the overall spread of the data, then we can't definitively say that these weren't drawn from the same distribution. This is what we would generally use if there are two categories. There's a related statistic called an F statistic, or used for analysis of variance, ANOVA. You usually see that term ANOVA associated with this, that we would use if we have more than two categories. So in an ANOVA test, we're basically doing the same intuition. We are looking at the variance of the means, so asking how far apart the means of our different categories are. So if, let's say, we had 3 alleles, C, G, or A at this site, then we would have 3 categories. We would compute the mean value of each category and ask about the spread between those mean values, the variance of those, which is more or less equivalent to taking the difference between the means in the basic 2-category test. More precisely, it's roughly comparable to the square of the difference between the means. And then we divide that by the mean of variances. So we're controlling, again, for the fact that a large difference between the means of the categories is not significant unless there is a relatively smaller value of variation within the individual categories. So those are the basic statistics we would use. And we can visually see why those would work by looking at a few examples here. So imagine that what we're seeing here is a number line where we've plotted, running left to right, let's say, the yield of ears of corn from our corn plants, and where the different colors correspond to different allele values. So in this first case here, up at the top, I'm going to say that maybe the G allele is in red here, And we have a very low weight here, somewhat higher, so a bunch of values of weights from G allele plants. And then maybe the C allele is in blue here. So we have a bunch of different weights of plants that have the C allele. And to compute our t-score, what we would do is compute the mean of the values within each category. So we can say the mean of the red values is approximately here, the mean of the blue values is approximately here, and we would say that the distance between those is the distance between those two mean values. We then want to know if that is large relative to the spread within the values, so we would compute that variance, which is a statistical measure of the spread, averaged between the two different categories, and we would end up with something roughly on the order of this region here. A little bigger maybe than the distance between the means, and so we would say, looking at this, that the ratio is not very big. Even though there looks like there might be an appreciable difference between the means, it's not large relative to the square root of the variance within the categories. So this probably is not a significant association. In the second case, if we compute the means of the red and blue categories, we get about the same difference as in the first case. So our numerator would be about the same. But our denominator is much smaller. We have much tighter packing of the points within each of the categories. So we would end up concluding there is a very small variance within category. And so the ratio of the difference between the means and the square root of the variances is going to be much higher in this case. And the second case would look like, very likely, convincing evidence that these are not drawn from the same probability distribution, and that would show up as a low p-value for our t-test. So I should point out that with the t-test, we're usually doing a two-sided test, but on either side of this, you would be looking for a low p-value, so low probability of that ratio of numerator to denominator being as skewed away from 0 as it is. In the analysis of variance case, the de novo, we might have a data set like this. This would be showing 4 alleles, maybe corresponding to different haplotypes of interest to us. And we saw data like this. We would compute the means within each of these categories. We would measure the variance between those 4 mean values, which would give us something that would be approximately on the order of that distance. I'm mapping out here squared. We would compute the mean of the variances. Each individual variance is a much smaller value because within each category the data points are very tightly packed. And so we would end up with that analysis of variance X statistic showing a value where the numerator is large relative to the denominator, and we would hope that that would then give us a significant p-value. So a small p-value that would indicate that whatever category we're looking at here that gives us these 4 different values, whatever variant is associated with the phenotype we plotted on this number line. So that basically is statistics for quantitative trait loci. And these bring up many of the same issues that we would have to deal with when working with case control association, that we would also typically be looking at a large number of variants, maybe a whole genome, and have to worry about these issues of controlling for multiple hypotheses through von Fermi correction or other designs. So there's a lot of recent research into these questions for both QTLs and the categorical association testing. A lot of this going into more advanced ways of doing the statistical testing to try to squeeze a bit more power to find those relatively weak variants out of the studies, entirely new methodological directions. So a lot of work coming out of the field of machine learning, something we'll be hearing about more later in the term. So basically, for other ways of trying to learn models of these associations that might be able to discover effects that would be very hard to find in these more traditional study designs, better ways of speeding up the cost and or lowering the cost of conducting these tests, and a lot of work of gathering large data sets, so genotyping large numbers of species that are important to us and large numbers of individuals, resequencing them within these species. There is also a lot of work in more of an early stage trying to bring these kinds of things into clinical work through what's called personalized medicine that I mentioned a bit earlier, trying to figure out how best to use this kind of information about genotype and phenotype correlations, these risk models, to improve the way we diagnose and treat patients and make recommendations for them about how best to avoid or be treated for diseases, as well as pharmacogenomics, other ways of bringing this into treatment practice, for example, by adjusting what drugs are given to a person or what other kinds of treatment based on knowledge of how they're likely to respond from their genetic traits. So a lot of this is very current research, things people are trying to do right now. And I mention this in part because I know many of you taking this class are pre-meds or hoping to be working in medicine or in basic biological research that may contribute to medicine. And I think there's a widespread belief within the field that this is a big thing that is beginning to change and is really going to dramatically change the practice of medicine for both the researcher and the doctor working in the clinic. So this is something you're not traditionally going to learn a lot about yet in a pre-medical or a medical education, but it's very likely in the future you're going to need to know this, so be aware of this if you might be working in those directions. All right, so any questions about any of this? Okay, so I guess we'll stop there.